This tutorial is to introduce the student to the gene details page that is part of the Integrated Microbial Genomes Annotation Collaboration Toolkit. And as with all assignments in this case, we are going to begin by logging in. I have a bunch of different accounts, so I need to, in fact, select my appropriate student. My name is Test Student 85. So as with the previous tutorial, recognition here is that you're given an assignment, and your assignment is in fact assigned to a student team. In this particular example, you are a team of 1. I'm going to open my assignment and see that 3 genes have been assigned to me. And in each particular case, these genes have a unique gene object identifier number. They have a laboratory notebook, a lab report, and a blank that will be filled in by the student in a second. So in order to get things going here and remind you that I like to open my links in new tabs, I'm going to open the gene object identifier link, which opens a gene detail page. The gene detail page is where you'll be going for quite a bit of information in your basic information module, in your structure-based similarity module, and your sequence-based similarity module, and it's sort of a home base, so to speak. So just to take you through a brief tour of this, we'll notice that again, this is a particular sort of vital statistics page with an enormous amount of information that is really information overload for all but the most distinguished and serious bioinformatics scientists. We're just going to take a stroll down this page until we get to the bottom and see that there are lots of different tools available for somebody who is particularly interested in any kind of gene. Recall before that this was listed as a gene that's in the minus strand. So we often talk about genes reading in the left to right direction up on top here, but you need to recognize that genes can be read in the right to left direction as well. Getting back to the top, I'm just going to point out some of the features of the gene detail page that you will need to be able to complete many of your assignments in this class. So first of all, there is a number here, a gene ID that has been assigned to this particular part of the genome. There's also something called a locus tag. There is a predicted gene product name that the machine has generated. There is the organism the genome came from. There are the DNA coordinates, which represent the minus strand, something that's 1,077 base pairs long. This is a piece of the piece of broken DNA that the annotation came from. The GC or guanine cytosine content of the genome, and all kinds of other pieces of information, including the amino acid sequence length. We're going to go here, and the peptide sequence length, another place we'll be going later on in the day. As we move on down, this is what I call the gene neighborhood. It's a very amazing mechanism for us to see how genes are potentially organized within the genome, and especially the idea that genes of similar function are organized and regulated in similar manners within the context of an entire genome. So one last piece that will pop in here is this idea that this gene detail page is a bunch of gene information that was pulled from the entire genome of an organism called Thiomicrospira crunogena, an organism that was identified, a microorganism identified in a deep sea vent. So by opening this link in a new tab, I can see some vital statistics about the entire organism. You won't be asked to know these vital statistics, but this is just sort of, again, an interesting place to start looking. You can see the name, the proposal name, and the name of the organism, both genus and species. You can also note that the location, the geographic location where this organism was isolated was a deep sea hydrothermic vent. And there is a publication about the isolation. This is of this organism. This is true discovery science. Someone rode in a deep sea submarine, went down there, and isolated this, a chemolithoautotrophic bacteria that is a member of the proteobacteria, gamma proteobacteria, Thiotrichales. I can't even say the family name here. The point is that we are in a genetics class, but I don't ever want to lose touch with the fact that we are studying all of life. And here we go. A mistake. Come on. Let's see if we can't get it back. No, I'll stop and reload the page. Well, I'll just stop the recording right there.